Am I audible, visible? Am I audible, visible? Hi everyone, just let me know, am I audible and visible to you all? Quickly in the chat section, I'm waiting for your comments. Hi students, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. Welcome to this one shot of principles of inheritance and variation. So it is going to be a quick revision session for all of you. You know that in detail we have already discussed this chapter. We have a separate playlist link for this particular chapter. So here we are going to revise it. And what is the reason behind revision? You know that it's Saturday today. Tomorrow it's Sunday. I'm not taking any of the classes right on the YouTube, but definitely on the Unacademy platform. So bache, you have the time to revise this chapter. So genetics, physiology, and your ecology, the unit reproduction, biotech, you know that these are the unit from where many questions used to come. So let's revise them again and again. Okay, so whenever I'm going to get the time, that's what I'm going to do. We are going to take the revision sessions. Okay, right, Sabarish, we didn't start the session yet, and you are asking about the number of hours. Not fair, not fair, not fair. So everyone quickly hit the like button, and if you're new to our channel, you know what you have to do. Just hit the subscribe button quickly. If you are new to our channel, quickly hit the subscribe button and be the part of this family. And trust me, we are going to bring so many sessions and where your teachers are going to be like this, enthusiastic. Okay, that's how we are going to teach you. So quickly hit the like button, everyone. So here you guys can see my videos, right, other teachers videos. This is the video section, so you know that time to time we keep on guiding you people, right? We provide you the information which is important for your NEET examination, right? Okay, so mutation, bache, Karthik, even we have discussed in principles of inheritance and variation, and a part of it we also discussed there in the molecular basis of inheritance. Don't worry, today let's discuss about that too. Okay, and if there is any topic, bache, that you want specifically, okay, that you want that ma'am you should teach this topic in detail, guys, please put the topic name in the video section, in the comment section. I will pick up that comment section. Okay, I will pick up the questions from comment section. I will make the video accordingly. And tell me which other video you want, which other session you want. Like you want me to take to make videos on the strategies, on the strategy based session, on the syllabus, how to prepare for the NEET. What do you want? Tell me. These days I'm not so active on Telegram, otherwise I'm going to put it there, right? So you tell me quickly that which particular topic you want me to cater. I will make the video on that, and we can even have a live session. What's it? Okay, so what you have to do, if you want guidance, if you want same type of sessions, now you just need to do one thing, students, and that is, see, not this song, of course. So here you can see, you can check any of my video. Fine, you can check any of my video, and in the description box here, see, the link is given for my batch. That is your Avengers 3.0 batch, and currently it's unlocked 25%, so you are getting the additional discount. It's just that you have to use one coupon code, that is Ambika10. Okay, and you will be the part of my, yes, you will be the part of my batch, where you are going to get doubt sessions, you will get the discussion sessions, the regular classes, the test as well, and as per your demand, bache, I used to keep the sessions. Okay, so if you want to be the part of this amazing batch, here you guys can see the coupon code Ambika10 is already applied, and you will get this batch at a price of 3,000 rupees, 3,157, just 3,157. Okay, so be the part of this batch and boost your NEET preparation. So as one student is saying that, ma'am, we want the one-shot lectures on ecology, done. It will be done. What else I can do for you? Just let me know in the comment section, bache, so I will do it accordingly. Okay, don't worry. So here you guys can see, as I said, bache, this is the coupon code that you have to apply. The coupon code is Ambika10, and whatever batch you want to join across the Unacademy platform, you will get the lightning deal over that. And especially join the Avengers 3.0 batch, because here in this batch currently we are revising the class 12th syllabus, and then we are completing the class 12th syllabus, and then we will start your class 11th revision as well. Parallelly class 11th revision is going on. Here you can see this. Okay, here you can see this. So go to the download the Unacademy app. You can search my name, right? You have to subscribe my profile, or directly on Google you can type Unacademy, right? You can search my profile, and this is what you have to do. You have to subscribe my profile, bache, and the classes that I'm taking now, you will get the detail of that. See, my batches, my free classes as well. Today we discuss plant growth and development. It was the first class on plant growth and development that I have discussed today. So you will get the many courses here, and you have to pay nothing for that. So be the part of these classes. Okay, be the part of these classes. So now let's move to our main topic, that is the principles of inheritance and variation. So it is going to be a revision session for all of you, and that these are the topics that we are going to cover today. Okay, we'll be talking about the laws, we'll be talking about the inheritance of one gene, inheritance of two genes, the sex determination, mutation, and the genetic disorders. Clear, bache? That's what we have to do. Let's start the topic. Let's start with the basic introduction. Okay, I will just wrap it up. Okay, so you tell me about the genetics. That what do you understand by the term genetics? Quick, it's a one shot. It's a one shot, so I'm going to increase my speed as well, but I want responses from your side too, guys. So please, in the chat section, be active and answer all my questions. Answer all my questions. And see, you can see one Nandini here. She's a very regular student. So Nandini, do let me know in the chat section here your experience of the special classes. Quickly, do let me know about your experience of the special classes here, Nandini. That's a request. What is the genetics? Quick. What is the genetics? Quick. Yes, you are right. When it comes to the genetics, it is the collective study. Collective study of what? It is the collective study of the heredity and the variations. That's what we are going to study here in the genetics. Right, bache? So when we talk about the term genetics, what we discuss? When we talk about the term genetics, it was given by William Wedson. Right, it was given by William Wedson. He gave the term genetics, and actually in the genetics we study the heredity, we study the variation. If you read it from NCERT, I am sure that you know about this line that like begets like. I hope you all know about it, right? In that first paragraph, right, of the starting, when you study this chapter from NCERT, this is the line that you find, right? Like begets like. Now, what is the meaning of this line? Like begets like. See, bache, when you talk about the human beings, obviously human beings are going to give birth to the young ones, and that young ones look like the humans. Or obviously, even in the case of elements, when they are going to give birth, obviously in the case of elements, what is there? Elephants, what is there? Their young ones also look like them. You know it, isn't it? You know it, isn't it? Even in the case of dogs, even in the case of cats, obviously when you talk about their next generation, it is going to be same like the parent generation, and we know it very well. So that's why we say like begets like. And why and how is it possible? It is possible because of the heredity. So what is the heredity? It is the transmission. What is the heredity, students? It is the transmission of characters from one generation to the another, or you can say that transmission of characters from parents to offsprings, isn't it? From parents to offsprings. That is what heredity is, isn't it? So our parents are going to give us the information. Our parents are going to give us the DNA, the genes, isn't it? Guys, is there any issue with the... Is there any issue with the voice, guys? Is there any issue with the voice? Thank you so much, Nandini. You all can read the comment. Nandini's comment. Is there any issue with the voice? No, it's perfectly fine, Trisha, Nandini. It's perfectly fine, right? Okay, so like begets like. Why is it possible? Because of heredity, the transmission of characters from parents to the offsprings, right, bache? And many times we confuse these two terms, heredity and inheritance. I hope you know about the inheritance as well. So inheritance is actually the process of that transmission. When in general I'm saying that characters will pass from parents to offsprings, that is heredity. But I'm saying that the process of, right? In general, you are saying I'm doing this, or you are saying that is how I'm going to complete this task. These are two different things, isn't it? So when it comes to the inheritance, it is the process of transmission of characters. It is the process of transmission of characters from parents to offsprings. Done, bache? So that's the difference in these two words. First of all, understand it. Now, when it comes to the variation, how will you define variations? Tell me quickly. How are you going to define the variation? Bache, yes, these classes are more than sufficient for you. Yes? How are you going to define the variations? See, we all are human beings, right? We all are human beings, but still, right, we are not same. We are similar, but we are not same. We have the degree of variation. Our features are different, right? Right, the differences can be on the basis of the height. It can be on the basis of the eye color, features, general morphological differences are also there. Like basic form is same, but look wise we are not same. Right, we are not same. So when it comes to the variation, so here what are you going to discuss? That individuals of the same species, right, they have a degree of they have the differences, okay, and that differences are basically the variations. Clear, bache? That differences are basically the variation. So individual of same species, they have some differences, and that is known as variation. I'm writing it here. Individuals of same species. Individuals of same species are different from each other. You can say that they have individual of same species have differences, and that differences or the degree of that differences is known as variation. And that differences are variations. Can you tell me what are the reasons behind variations, and variations are good or bad? Quickly tell me. But Karthik, my audio is perfectly fine. I really think that you need to start your system. Kindly do that. Yes. How are you going to define the variation? Tell me quickly, students. How are you going to define the variations? Variation means changes, and we need that changes, right? We need that changes for the survival, to adapt in the different environmental conditions. That's what you need to keep in your mind. Students, when we talk about the sexual reproduction, we discuss the meiosis, because meiosis is the division, right? Meiosis is something which will form the gametes. And when it comes to the sexual reproduction, you know that gametes, right? When we talk about the sexual reproduction, you know that gametes, right? They are the haploid cells. That haploid cells will fuse and they will form the zygote, and that zygote will further grow. That's what we all know, isn't it? So why meiosis is important? Why sexual reproduction is better than asexual reproduction? Because it results in the variations. How? Because of crossing over. Because of crossing over. So when we discuss crossing over, okay, when we discuss what? When we discuss crossing over. So crossing over is going to do what, bache? Because of crossing over, maternal and paternal chromosomes, they will exchange their segments, isn't it? They are going to exchange their segments, and right, in the next generation, but different characters will be there. Like I have the characters of my father as well, of my mother as well, isn't it? Isn't it? They are two different individuals, but I am having the characters of both of them, right? Right, so it's something different. So how is it that possible? It is possible because of sexual reproduction. It is possible because of meiosis. It is possible because of crossing over, right, bache? So variation. So one reason for variation is the crossing over indeed, and another thing is the mutation. So now in the evolution chapter as well, we talked about the mutations, isn't it? They are the sudden changes, sudden heritable changes they are. Okay, so these two things are responsible for variations, and variations are important to adapt in the changing environment. Done, bache? Adapt in the changing environment. Clear, bache? So this is about the basics of the genetics here, and now you know about the principles of inheritance as well as the variation. Nandini, Siri, Subhashini. There are two students, those who are again and again saying that there is some issue with my voice. Is it so? If it is so, do let me know. If it is not, then I will ignore these messages, of course. Abhisha, is there any issue with my voice or it's perfectly fine? Quickly tell me in the chat section, please. Like, write it like this, na? No, ma'am, there is no issue. All clear. Are you sure? Are you sure? So should I ignore that messages? Should I ignore that messages? Okay, so this is about the genetics. So when you talk about the father of genetics, let me write it here. So father of genetics is Gregor John Mendel. Okay, Gregor John Mendel, he is the father of genetics. And bache, when you talk about the father of experimental genetics, right, these two things are important. When you talk about the father of experimental genetics, then the name is, anyone here in the class? That's Thomas Hunt Morgan, T.H. Morgan, right? Thomas Hunt Morgan, T.H. Morgan. Is that clear? That's T.H. Morgan. Is that clear? So that's what you need to understand initially. Fine, bache. So now let's start the story, and let's start the story, of course, with the Mendel, right? Let's talk about the Mendel and about some basic terms. Okay, so I am not going to write their definitions. Okay, we are just going to revise it. Let's start. So if you want to understand the if you want to understand the genetics, yes, you need to know about certain terms. Now, what do you know about the factors? Do you know what are the factors? Do you know that? What are the factors? Quickly, what are the factors? Anyone? Yes, Bache Mishra? Anyone? So it is the unit of inheritance. Factors are what? It is the unit of inheritance that is responsible for what? It is the unit of inheritance that is responsible for the transmission, isn't it? Isn't it? What are the factors? They are the unit of inheritance that is responsible for the inheritance, right? That is responsible for inheritance and the appearance of a character, isn't it? And the appearance of a character, that's what a factor is. But say these days we do not use the term factors. Instead of that, we used to call them as genes. We used to say now that genes are the basic unit of heredity. And where do we find these genes? We find them on the your DNA, right? So now let me start this part in a different way. Okay, in a different way, let's talk about it. You can take the example of anything. You can take the example of a prokaryotic cell. You can take the example of a eukaryotic cell, whatever you want, right? Any example you all can take. You can take the example of a prokaryotic cell or even the eukaryotic cell. So when you talk about a cell, so in the cell, what do you have? You have the nucleus, right? In a cell, what do you have? You have the nucleus if it is a eukaryotic cell. And even if it is not a eukaryotic cell, then again, DNA is there, right? DNA is there, right? Am I right? So when you talk about the nucleus, so nucleus is having the genetic material that is DNA. And when you talk about this DNA is having the basic unit of heredity that is known as gene. But now we use the term gene. Initially, right, Mendel called these called it as factors, right? Mendel called it as factors. So you know that the DNA is present in the nucleus and that DNA is having the genes, and that genes are the basic unit of heredity, right? They are the basic unit of heredity. Clear, bache? Clear, bache? Now, so before understanding genetics, let's talk about the Mendel, and then I will tell you about the basic terms. So Mendel is the father of genetics. I told you the story as well, right? So why? So Mendel is the father of genetics. What Mendel did? He conducted hybridization experiments, right? He conducted various hybridization experiments, and the one he conducted on the pea plant for the 7 years, that experiment was the successful one. Clear, bache? Now, when it comes to the Mendel, what is the reason behind his success? Because he added mathematics to the biology. And for the first time, someone used such kind of approach. Okay, for the first time, someone used that kind of approach where they are using mathematics. Okay, mathematics in the biology. And that is the reason at that time, right, when he published his work, or his work didn't get recognition when he was alive, but after his death, 3 scientists, they rediscovered the Mendel's work. First of all, let me tell you the story. And let's start with the story building, and then we will talk about this concept. So when it comes to the Mendel, he belonged to a very he belonged to a very basic family, right? So he wanted to be a teacher as well. And at that time, I was reading about Mendel. So at that time, you know, some exam need to crack if someone wants to be a teacher. So there was a story like this. But finally, you know that Mendel was a priest, right? He joined a monastery, and there he studied as well. Right, there he studied as well. So he added whatever experiments he conducted, he applied that probability on that, right? He calculated it basically. So mathematical approach was there. And because if you talk about the Mendel's childhood, right, the family was like this, that he had that interest in the natural sciences, in the biology, in that, like, he was connected to all that things. Okay, he was connected to all that things. So that's why he experimented like this. So finally, he studied a lot, right? I will not tell you that things in depth right now. So the successful experiment, Mendel's hybridization experiment, again I'm repeating, that he conducted for 7 years, right? Because of that, we got to know about certain basic rules of inheritance. Okay, about certain what? About certain basic rules of inheritance. Are you getting it? Are you getting it? So that's why we consider Mendel as the father of genetics. And as I said, he didn't get the recognition when he was alive, but after his death, 3 scientists, they rediscovered Mendel's work, and then he got the he got the recognition. So posthumously means after death he got the recognition. And 3 scientists that rediscovered Mendel's work, they were, let me write it. 3 scientists, they rediscovered Mendel's work. What they did? They rediscovered Mendel's work. And their name is your Hugo de Vries. He even told us about the mutations, right? Carl Correns and Erich von Schirmack. Okay, Erich von Schirmack. Are you getting it? These were the 3 scientists, they rediscovered the Mendel's work. That's what you have to remember. Now, let me start with the terms. So as I said, instead of genes, Mendel used the word factors, right? Mendel used the word factors. So he considered factors as the basic unit of inheritance. He considered factors, these factors, they occur in pairs, right? They are the discrete units, but they occur in pair. That's what, right? That's what he did. Fine, that's what he did. He called them as what? He called them as the factors. What he called them, bache? He called them as the factors. So now, as I said, that you need to know about the certain terms, and then this chapter will be easy for you. So now you know that instead of factor, we use the term gene. So you are well aware of it, right? So genes are going to be there in a pair. Okay, genes, they are present in a pair. Let me tell you why. Okay, so you all know about the chromosome, right? You all are well aware, you all know about the chromosomes, but at that time, that things were not so clear. In this chapter itself, bache, we will be discussing all that things. Okay, we'll be discussing that how do we link Mendel's laws with the chromosome part, with the chromosomal segregation part. Okay, in this chapter we are going to study that. But as of now, you know these things, isn't it? We know that when it comes to the humans also, right, we are diploid organisms. We have 46 chromosomes. So basically we have 23 pairs, right? We have 46 chromosomes, means we have 23 pairs. So one chromosome is from the mother and another chromosome is from the father. One chromosome is from the mother and another chromosome is from the father. That's what we know. Yes or no? That's what we know. So now imagine, bache, see, these are the chromosomes. One is from mother, one is from father. You know that we discuss about the homologous pair of chromosomes, isn't it? We talk about the homologous pair of chromosomes. So what is the meaning of homologous pair? Bache, as I said, we are diploid. right? Our chromosomes are in pair. So if I'm talking about the 46 chromosomes, 23 chromosomes are given by mother, 23 chromosomes are given by father. So that's why I'm saying that we have a pair of chromosomes. Are you getting my point? Right. So when we use the term homologous pair of chromosomes, homo means same. What is the meaning of homo? Homo means same. So the chromosomes having same centromere position, the chromosomes having same centromere position, same size, okay, the chromosomes having same centromere position, same size, okay, same genes, same size, same shape, same genes, they are the homologous chromosomes, right? Like here, just imagine, right, just for an example, let's say here at this point, if there is a gene, if there is a gene for height, let's say there is a gene for height in the case of mother, here also in the father, when it is a homologous chromosome, here also there will be a gene for the height. That's why we say that genes, they occur in the pair, because even our chromosomes are also there in the pair, but they studied it later, right? It was not discovered by them. Fine, it was not discovered by them. So genes, they occur in the pair, and we used to call them as alleles. This is another term that you need to understand. What we used to call it? Yes, Bacha Abhisha. What we used to call it? We used to call it alleles, right? So what are alleles? They are the alternative form of a gene. What are alleles? They are the alternative. They are the alternative form of a gene. What are alleles, everyone? Just type it in the chat section quickly. The session should be interactive, so all of you, please type it in the chat section as well. So what are alleles? They are the alternative form of a gene which are located on same position on homologous chromosome, right? Which are located on same position, which are located on same position on a homologous chromosome. Now you know the meaning of a homologous chromosome, and I told you that if there is a gene for height in the case of mother at a particular position, right, at the same position in the case of father also, you will find the same gene, but its alternative form can be different. Its alternative form can be different. Now let me explain this. So I told you genes, they occur in pair, right? So basically a pair of allele used to make the genes. So as I said, what are alleles? They are the alternative form of a gene, right? They are the alternative form of a gene, like the variant of a gene it is. It's a variant of a gene. Let's say there is one particular gene, if it will get mutated with time, okay? How these allele forms? They form because of mutations, because of sudden changes. So if this allele, if it will get mutated, you will get it some another form. You will get some another form of it. Okay, so that particular form is that allele. Okay, it is the alternative form. I'll give you one example. I'll give you one example here. And with the help of that example, I'll even explain the character and trait to you. So if you understand these terms now, then everything will be easy for you in the chapter genetics. So whenever you are going to start the principles of inheritance and variation, you should know about these terms. Clear, bache? You should know about these terms. Done? So let's take one example. Let's say height. Bache, height is a particular character, isn't it? What is height? Height is a particular character. Yes or no? Yes or no? So height can have two forms. A plant can be tall, a plant can be dwarf. A plant can be tall, a plant can be dwarf. In the nature, you will not find all the plants as tall. You will not find all the plants as dwarf. Some are tall, some are dwarf. Some are tall, Subhashini, and some are dwarf. And sometimes, bache, not sometimes, and this is what we also observe, that certain characters, they are dominating. You will find them more. Like if I talk about the eye color, in India, very rarely you will see bluish kind of eye color. Isn't it? Isn't it? Right? We do not have that. We do not have that. Right? You can compare ourselves with the people of another country. So you know that, right? Differences are there in our skin tone, right? Even in our hair color, eye color. Why? Why is it possible? Because of the genes, of course. Because of the genes, plus environment is also going to play the role. So there are certain characters, even if you talk about the Indian people, so we have certain characters which are quite common, and there are some characters which are rare. which are rare. They are not, you will not see such characters so frequently. You know it very well, right? So same is the example here. Height is a particular character, right? But an individual can be tall, an individual can be dwarf. So these are two variants of the height. These are two alternative forms of the height. Yes or no? Or even I'll give you one example in the context of a phone. Let's say you're going to buy a mobile phone. So tell me which mobile phone you want to buy. First of all, tell me which mobile phone you want to buy. Quick. Tell me. Arey, you can tell me your dream mobile phone as well. Wow. As expected. है ना? iPhone, ma'am, iPhone, as expected. अच्छा, any Saiti is like, ma'am, any smartphone. है ना? iPhone, okay. So which one? iPhone 15. iPhone 15, है ना? Everyone is like iPhone, iPhone, iPhone. चलो ठीक है. Let's say today you have 2 lakhs in your pocket and you are like that, ma'am, I'm going to buy a phone. So you will check the variants. You will check whether it is having 256. What? Storage you are going to check, na? Yes or no? The storage you are going to check, na? Yes or no? You people are going to check the storage there. You will check the different variants of that storage, 128, 256 and that 512 if I'm not wrong. है ना? So different forms they are. So basically it is your iPhone. Basically it is your iPhone 15, but it is having different variants, different forms are there. Same as the example here. Height is a character, but that height is having the different variant. The tallness, the dwarfness is there. But in the case of pea plant, let's take the example of pea plant. You will see more tall plant. So this character is going to be the dominant one. So which characters are dominant? Yeah, which traits are dominant? The one the characters, the traits which we see more, which express them more. Clear, bache? They are the dominant one. And the one which is not so expressive, that will be the recessive one. So here, when I'm saying height is a character, these are two alternative forms, right? So they are the traits. They are the traits. Right? So height is a character, tallness and dwarfness is a trait. Same as the example here. So gene is responsible for this character. A gene is controlling this character, but its alternative forms, right? That are the alleles. So that alleles are going to decide that whether the individual will be tall or the individual will be dwarf. So that's how you have to understand it, right? That's how you need to understand it. So the characters which expresses itself too much, right? The characters that you will see in majority of people, that character is the dominant one. Okay. And for dominant characters, how are you going to denote dominant characters? With the help of capital T. And when you talk about the recessive characters, which characters are recessive, which are not so expressive? Okay. And you are going to denote it with the small letter. Okay. So capital letter is for the dominant characters, small letter is for the recessive character. Okay. So alternative form of a character is what? That is a trait. That's what you need to remember. And that's how you need to understand the gene and the allele as well. Now the next term is, bache, you know that we are diploid. Right, we are diploid. So on the basis of that, I'll be explaining two more terms, that is homozygous and the heterozygous. Right, homozygous and the heterozygous. Homo means same, bache. Hetero means different. Homo means same, and what is the meaning of hetero? Hetero means different. So you have two conditions here. See, homozygous dominant, homozygous recessive, and heterozygous. These are the three terms that we have. So when it is the homozygous, homo means same. Now you know that we are diploid. You know that we have the pair of chromosomes. You know that our alleles are going to be there in the pairs. Yes or no? You know that our alleles are going to be there in the pairs. Right, bache? Right, bache? So let's say you are talking about a plant. If in that plant, both the alleles are dominant one. First of all, homo means same. If both the alleles are same, homozygous condition is there, because both the alleles are same, Priya, and both the alleles are dominant one. So that's why I'm calling it as homozygous dominant. This part clear? Next, homo again means same, homozygous recessive. Homozygous recessive. So homo means same. Both the alleles are same here, right? And they are the recessive one. So it is going to be the homozygous recessive. Now the term is hetero, but hetero means different. What is the meaning of hetero? Hetero means different. So both the alleles are not same, they are different. So this is going to be the condition. So Nandini, if we have one capital T and one small t, which trait will express? Which trait will express? If we have one capital T and one small t, which trait will express? Obviously the tallness. So here the individual is going to be tall. The individual is going to be tall. So always remember, when the condition is heterozygous dominant, always remember when the condition is heterozygous, right? When the condition is heterozygous, so it is going to be dominant as well. Why? Because there is the presence of one dominant allele. So even if in that pair, one allele is the dominant one, then definitely dominant character will express itself. Then definitely what is going to happen? The dominant character is going to express itself. So these three terms clear? Yes, bache, these three terms clear. So the next term is what? Next term is the phenotype and the genotype. Of course, this is going to decide that. Next term is what? Next two terms are phenotype and genotype. So you should understand it, and then the next part will be easy for you, right? So what we have discussed so far, we started with the basic introduction of genetics, right? Then we revised about the Mendel, who rediscovered his work, and then we talked about the factors, about the genes, about the alleles, about these terms. Clear, bache? About these terms. Done. So now phenotype and the genotype. Phenotype, pH, physical, right? Phenotype, pH, physical. So here just keep one thing in your mind, that is physical appearance. Here just keep one thing in your mind, and that is what? That is the physical appearance. What is it, students? It is the physical appearance. Now I'm getting one question, let me answer it. One student is saying, can recessive be heterozygous? How is it possible, bache? Hetero means different. Hetero means different. That in a zygote, you have two different types of alleles. Or you can say that in a chromosome, in a homologous pair of chromosome, you are having two different alleles. So if two different alleles are there, if by any chance, if there is the presence of the dominant allele, if by any chance there is the presence of dominant allele, then 110%, 110%, you will see that the dominant character will express itself. So heterozygous can never be recessive, right? Recessive is the one which is not expressing itself in the presence of dominant. A recessive trait will only be expressive when both the alleles are recessive. Okay, when both the alleles are recessive, then a recessive trait is going to express itself. So heterozygous is going to be the dominant always. Is that clear? Okay, so phenotype here, the word is physical appearance. What is the word here? The word here is the physical appearance. That's how you have to remember. When you talk about the phenotype, you are talking about the external, the physical appearance, right? This is what you are discussing when you are talking about the phenotype. You are talking about the external and the morphological appearance of an organism for a particular character. You are talking about what? You are talking about the external or morphological. External or morphological. Fine, you're talking about what? You're talking about the external or the morphological appearance of an individual for a particular character, right? Of an individual for a particular character. I'll explain it. Just allow me a minute. And then comes the genotype. So here you are talking about the kind of, here you are talking about the genes, right? Here you are just talking about the external appearance. I'll give you one example. Let's say there is a plant. I know my plant is looking very bad, but okay, try to manage. So you are looking at this plant. Fine, you are looking at this plant. By looking at this plant, you all can say now that whether it is tall or dwarf. You can comment, isn't it? Yes, Bache Venkat, you can comment. Of course, you can comment that this plant is tall or this plant is dwarf. So you just looked at it and you said, oh, wow, the plant is tall. So here, what are you discussing? The phenotype, the external appearance of a particular character, right? You are discussing about the phenotype. But see, if you are talking about the tallness, two conditions are possible. That plant can be homozygous tall. That plant can be heterozygous tall. Yes or no? That plant can be homozygous tall. That plant can be heterozygous tall. Homozygous tall means the genotype will be this, capital T, capital T. Heterozygous tall means the genotype will be capital T and the small t. Nandini, crazy, it will be capital T and the small t. So in both the cases, the phenotype is going to be same, but the genotype is different. So when we talk about the phenotype, we are just talking about the external appearance. We are just talking about the external appearance. But when we discuss the phenotype, okay, when we discuss the genotype, you are talking about the genetic makeup. Okay, you are talking about the genetic makeup. Okay, so that's the perfect example, homozygous tall or the heterozygous tall. That's how you can remember this. One more thing, always remember, phenotype is the result of genotype. Our genetic composition is going to decide our morphological appearance. But yeah, here even environment plays the role. Okay, environmental factors are also there. Done, bache? So this is about the phenotype and about the genotype. So tell me, this part clear? The basic terms clear? The basic terms clear? Sure, sure, sure. Are you sure? Sure, bache? Done. So now let's start talking about the Mendelism. So you know about the Mendel. The name is Gregor John Mendel, right, bache? So he was a priest and he started his experiment, his historical experiment on pea plant in 1856 or near about 1857. So he conducted that experiment for how many years? For the 7 years. For how many years? For the 7 years. So this is a PYQ. So from 1856 to 1863 he worked, right, bache? Right, bache? So he worked from 1856 to 1863. So the laws that we discuss now, the Mendel's first law, second law of inheritance, the first and the second law of inheritance, the laws that we discuss, bache. So that is not something that, you know, he concluded after 1 or 2 experiments. Imagine, he experimented for 7 years, right? He experimented for 7 years. Are you getting my point? Right? Then he concluded the things. So for 7 years he worked, and he worked on pea plant. He worked on what? Pea plant. That is your Pisum sativum. Fine, bache, Pisum sativum. But still, after working on the pea plant, after getting the result, he didn't get recognition at that time. I think about 16 years, or I don't remember that exactly. I think after about 16 years, he got the recognition for his work. Okay, he got the recognition for his work. Right. So basically, after 16 years of Mendel death in 1900, right, 3 scientists, they rediscovered his work and he got the recognition. And the name of that 3 scientists, of course, Hugo de Vries, because they were experimenting something like this, something like that. Right, they were when they were trying to, you know, decode all that rules for that inheritance, then they found that someone has conducted experiments before them. Imagine. And why? Because at that time people were not, right, people were not able to accept that how can someone mix mathematics with the biology. Okay, because of that thought only. Imagine, because of that thought only. Okay, so Hugo de Vries, Carl Cons, I don't remember that exactly. Maybe it is 16 years or the 30 to 33 years. Even I have to check, we'll let you know about it. So Eric von Schemark. So you have to remember the name of these three scientists. So they rediscovered the Mendel's work. Okay, so even he was, right, it was his good choice that he opted the pea plant. I'll tell you why. But before that, can you just tell me about the reasons that why Mendel didn't get recognition for his work? Anything that you think? Anything that you think? Tell me. Anyone? Exactly. You're right. You're right. Right? He tried to publish his work, and he tried to publish his work near about, you can say that in 1865, he tried to publish his work. But at that time, actually, the communication was not so easy. Okay. And moreover, he gave that concept that the genes, right, they are, you know, he didn't use the term gene, the factors are in pairs, right? They are pairs, they are moving together as a they exist as a pair, right? And they are the discrete units. They exist as a pair. Are you getting it? Are you getting it, Nandini? They exist as a pairs, right? But they behave like the discrete unit. They do not show any blending. They do not show any blending. But so at that time, scientists, they were not able to accept it. And yes, as you know, mathematics and biology, right? So that's why, right, no one was ready to accept his experiment, his work. Clear, bache? His work. Okay, okay. Yes or no? Yes or no? And what is the reason behind Mendel's success? Can you tell me? What are the reasons behind Mendel's success? Why he got successful result? Right. So his choice, his experimenting material, pea plant, it was a good approach. You know why? Because pea plant is having a short life cycle of near about 2 to 3 months. Okay. So after that, we can see the next generation, and the number of seeds, more number of seeds are produced by the pea plant. First of all, the life cycle was short, near about 2 to 3 months. And secondly, number of seed. So pea plant used to form many seeds. Okay, so it is easy to study the next generation. Okay, after 2 to 3 months, right? Life cycle is about for 2 to 3 months. So that's why it was easy to study the next generation. And moreover, in the pea plant, many contrasting characters were there, which is very important. Contrasting characters means the alternative characters. Like if I talk about the flower color, the violet, the white, okay, the flower position, the height. So many alternative characters were there. Many alternative characters were there. And moreover, pea plant is bisexual. So naturally, self-pollination is also possible, right? Naturally, self-pollination is also possible. Remember this trick, BMH. Do you remember this trick? BMH. Yes, BMH. That's my trick. So B stands for, from BMW, you have to make it BMH. Bisexual, monoecious, hermaphrodite. Bisexual, monoecious, hermaphrodite. Remember? Bisexual, monoecious, hermaphrodite. Right? So pea plants are what? They are bisexual. They are monoecious. So naturally self-pollination is possible, but and even in case if cross-pollination is required, so then what is going to happen? Emasculation, right? Even that's also easy. Emasculation, if they need cross-pollination. You know now what is emasculation? What is emasculation? Emasculation is the removal of the male part, means the removal of the anthers, right? Removal of the anthers. Clear, bache? Clear, bache? So, and even, right? So that's why he opted the pea plant. First of all, I'm telling you about the pea plant. Clear, bache? Clear, bache? Done? Done. Done. So first of all, the first reason is he opted the pea plant, and these are the benefits of opting the pea plant. This is the first reason. Secondly, if you talk about the Mendel success, right? बच्चे, Mendel applied mathematics, Mendel applied statistics, he applied statistical methods, right? And moreover, the large sampling size. Okay, his sampling size was quite large. Okay, the sampling size. Fine, bache. Fine, bache. So that are the reasons behind the Mendel success. And yes, one more reason is there. Like if you talk about the other people at that time, even they tried to study the inheritance, but they considered, okay, bache, Lakshmi, for bisexual, you use the word monoecious, bache. What are you talking for bisexual? You use the word monoecious, bacha. Bisexual flower means monoecious flower, of course. And unisexual is dioecious. And moreover, see, Mendel and his, at that time, the other people were also trying to study the genetics part, right, the inheritance part, but they were considering many characters at a time, right? They were considering what? They were considering many characters at that time, at a particular time, many characters they were studying. But when it comes to the Mendel, he studied the inheritance of one or the two characters at a time. Fine. He studied the inheritance of one or two characters at a time. So he got better results. Are you getting my point? So he got what? He got the better results. Isn't it? Isn't it? Done. So now let's talk about the pure lines. So you know about the pea plant now. You know that in the pea plant even the self-pollination is possible, even the cross-pollination, right? Self-pollination is possible, even the cross-pollination is possible, right? So first of all, Mendel conducted the experiment and he tried to form the pure lines. But when we use the word pure lines, means we are talking about the homozygous conditions, right? We are talking about the homozygous characters or the homozygous traits, right? Homozygous traits, as in, like, let's say he self hybridized the tall plant for many generations so that he can get homozygous tall, right? He hybridized the he basically hybridized the dwarf plant for many generations so that he can get homozygous dwarf. So first of all, he did what? He formed the pure lines. So pure lines means homozygous for a particular character. Clear, bache? Homozygous for homozygous, that they are having the homozygous traits. Clear, bache? Clear, clear, clear. So by continuous self-pollination, he got that. Okay, because of continuous self-pollination, he formed the pure lines. Okay, he formed the pure lines. And what Mendel selected? He selected 14 what? Many students, they make the mistake here. 14 what? Yes, what he selected? He selected 14 what? But it's not 14 generation. It's not 14 generation. Tell me quickly. Bache, he selected 14 true breeding plant varieties for his experiment. 14 what? True breeding. So what is the meaning of true breeding? Means pure lines, means homozygous for a trait. So 14 true breeding plant varieties. So basically he opted 7 characters, and all that characters were having the alternative forms, right? So that's why 7 characters or 14 traits. I told you about the characters and traits now. Like height is a character, but when you are talking about the tallness and the dwarfness, is what? They are the traits. Height is a character, tallness and dwarfness is a trait. Are you getting it? So that's why he selected 7 characters and the 14 traits. So 14 true breeding plant varieties means that plants are having that particular trait, and it is basically the pure line, right? It is basically what? It is basically the pure line. So you guys can see that 7 traits, 7 characters here. So which 7 characters he opted? The question can come from this part as well, right? So I'll start with this stem height, then we'll compare it with that picture, don't worry. Stem height, flower color, flower position, seed color, seed shape, pod color, pod shape, right? These are that 7 characters. Fine, bache. These are that 7 characters. Stem height, right? Then flower color, flower position, seed color, seed shape, pod color, pod shape. So even in the exam, they can also ask you that the characters selected by Mendel in that characters, how many characters were based on the color, how many characters were based on the shape. So question like this can come. Okay, question like this can come. So here you guys can see. So when you talk about the seed shape, which one is dominant? It is the round shape, capital R. So basically you will see the pea plant having round seeds, having round shaped seeds, right? So wrinkled one is the recessive one, small r. Then comes the seed color. So yellow one is the dominant, green one is the recessive. So that's what you need to remember. Flower color, the dominant one is violet and the recessive one is white. Pod shape, full pod shape, right? Full pod shape, and here it is the constricted one. Pod color, green is the dominant one, and yellow is the recessive one. So do not confuse seed color and the pod color. Pod color, and you all have seen the pea plant, right? So you know it well that pod color is green, it is the dominant one, and pod color yellow is not, of course not, right, bache? And then comes the flower position. So axial, right? One is axial, another is terminal. Axial means like this. Let's say here your flower is present, right? Not at the tip basically. So if here it is present at the tip, means terminal. But here, let's say if here it is present or here it is present, so it means it is axial. So axial one is the dominant character. So that's what you have to remember. And these are the 7 characters selected by Mendel. So see, they are the 7 characters. Of course, they are the 14 traits, isn't it? 14 traits. So he selected the pea plant variety which are homozygous for round shape, homozygous for wrinkled seed shape, right? Yellow colored for yellow seed colored, green seed colored, violet flower color, white flower color, right? Like this. Got it? Got it? Bache, I think Lakshmi, you are confused with the concept. It's BMH, bisexual flower. Bisexual flower means what? That it is having both male and female. So any plant which is having both male and female flowers, or you can say that any flower which is having male and female plant, male and female part, that is a bisexual or the monoecious flower. You are getting confused with these two terms. Bisexual means monoecious. Monoecious means that single flower or the single plant is having both male and female part. Hannah students, am I right or wrong? Tell me quickly. Am I right or wrong? Tell me quickly. So Lakshmi, I think you are confused. Bisexual and monoecious is having same meaning. Bisexual and monoecious is having same meaning. Sure? So now you have to tell me the chromosome number. Okay. You have to tell me what? You have to tell me the chromosome number where all that characters are present. And you know, Mendel was lucky because the character he selected, right, they were present on different chromosomes. In that regard, he was lucky as well. The characters he selected, na, it was the characters, these characters are present on different chromosome, right? Like chromosome number 1, chromosome number 4, chromosome number 5, and chromosome number 7. Chromosome number 1, 4, 5, and 7. Clear, bache? Clear, bache? Like seed shape is, the chromosome is 7th chromosome. So that's your homework, and I want to see it in the comment section, right? I want to see it in the comment section. So chromosome number 1, 4, 5, 7. These chromosomes are having these 7 characters. So I gave you one example, that seed shape. Okay, seed shape. This character is present on the 7th chromosome, and you will tell me about the others. Fine, you will tell me about the others. Done? So will you do it? Tell me quickly. Yes, so that's your homework. And same way, bache, as I said, see, be the part of these special classes as well. See, you have to subscribe my profile and subscribe it right now. See, the followers are 2K, they are very less. Let's make it 3K today. Okay, let's make it 3K today. So here you can check my batches and courses as well, where your Avengers 3.0 batch is there. Okay, you can even check my free classes here. Right, you can even check my free classes here. So here you can also see this. Today I started. Okay, these are my batches and courses. Avengers 3.0, of course, the NEET Energize batch. And see this, plant growth and development, even respiration done, etc, respiration plants. So we have discussed these topics in detail here, right? Not just the plant physiology, you will also see the questions also. You will see the genetics, DNA application, right? So be the part of these classes. Fine, be the part of these classes, and that's your homework that you have to do. So these are the 7 characters. So now the part that we need to start is that laws. Okay, let's talk about the laws of inheritance. So firstly, we'll be starting with the experiment and the results, and then we will talk about the laws. Clear, bache? Right, then we'll be talking about the laws. Okay, done. So first of all, I will tell you about the monohybrid cross, and then about the the conclusions, and on the basis of that we will discuss about the laws. So let's start with the cross first. Okay, let's start with what? Let's start with the cross first, and that is your monohybrid cross. That is what? That is your monohybrid cross. Clear, bache? So mono means a single, means single. Here what are you doing? You are considering a plant in which all other characters are same, but there is a difference of only one character. Right, what is the meaning of a monohybrid cross? So you are considering the inheritance of one character at a time. Right, you are considering here the inheritance of one character at a time. Like you are selecting the pea plant where rest all the characters are same, but there is the difference of only one character. Right, there is the difference of only one character. One question is there, that how can we say chromosome number by seeing character? Who's telling you that you can do so? Right, you cannot just look at the character and you can say the chromosome number. Of course not. Right, scientists, they have studied it, and it will be there in your book. You just need to check it out. You need to put Shri Divya, you need to put efforts to check it. That's all. You cannot just look at a character and you can say the chromosome number. Not possible. You have to study the chromosomes in detail, that sequencing and all, and it involves that sequencing part and all. Okay, so now come to this topic again. So inheritance of one character at a time. So first of all, you have to select the right parent generation. So when you are talking about the parent generation, firstly consider the pure lines. Okay, and then what will be there? You will do their selfing, their crossing. Okay, they are selfing, they are crossing. What is the meaning of the selfing and crossing? Obviously, you are hybridizing them, isn't it? You are hybridizing them, isn't it? So capital T means this parent is homozygous tall, this parent is homozygous dwarf, and then you will see the first generation, F1. F1 means first filial generation. F1 means first filial generation. Yes, bache, what is the meaning of F1? F1 means first filial generation, because one parent is tall, another is dwarf. So you know that this is what you are going to get. In first filial generation, all the individuals, right, all the seeds, they'll grow and they will form heterozygous tall plants, isn't it? Isn't it heterozygous tall plants? Tell me. Yes or no? All of them are going to form what? They are going to form heterozygous tall plant. They are going to form heterozygous tall plant. Done? Okay, so now after that, what we have to do, we need to cross the individuals of first filial generation so that we can check second generation, right? So that we can check second generation. And now, bache, to study that, to understand that, the Punnett square, we need to use what? We need to use the Punnett square. Do you know about the Punnett square? Do you know about the Punnett square? Right, bache, it is just like a, this is a Punnett square, okay? It is just like a checkerboard. It is just like what? It is just like a checkerboard. It is a Punnett square. It is just like what? It is just like a checkerboard, and we use this checkerboard to study the individuals, right? After a cross, right? When we want to study, now, what are we going to study here in this checkerboard? The result of the cross between two organisms, right? The result of the cross between two organisms that we have to study here. So this Punnett square is given by Reginald Punnett. Okay, so this is also a PYQ. Fine, this is also a PYQ. Clear, bache? So RC Punnett, right? Who gave it? RC Punnett. Fine. So it is a checkerboard to check the result of the cross between two organisms. Done, bache? So here, the first step, see, one is capital T, another, the two parents are having heterozygous, the two parents are heterozygous dominant here. So whenever you make a cross, we have to make the gametes first, right? You know why, right? Because you know that even in the case of human beings also, let's relate it with the human beings. So in our case, the gamete formation is there, the haploid cells, and that sperm will fuse with the ovum, and then there will be the diploid zygote. That's why we say we have the diploid number of chromosome, and or you can say that genes are occurring in a pair. Okay, genes are occurring in a pair. So whenever you draw the cross as well, right? So you have to consider the gametes first. Like, see, now the parent is having capital T and small t. So the gametes, because gametes, the gametes will be capital T small t, here also capital T small t. So when you consider this Punnett square, right, here, you have to consider both the sides. One side will contain the gametes of male, another side will contain the gametes of female. So it will be like this. So it will be capital T, capital T, capital T, small t, capital T, small t, and small t, small t. Isn't it? That's how you can check the result. So this capital T is what? This capital T means homozygous tall. Capital T, small t means heterozygous. Right? Heterozygous dominant means heterozygous tall. And here it is homozygous tall. And here what is it? It is homozygous dwarf. It is homozygous dwarf. Are you getting it? So let's talk about the phenotypic ratio first. First of all, let's discuss about what? Let's talk about the phenotypic ratio, phenotype. So one plant is tall, three plants are tall, one plant is dwarf. That's why it is 3:1. Okay, that's why it is 3:1. And then let's talk about the genotypic ratio. Okay, let's talk about what? Let's talk about the genotypic ratio. So one is homozygous tall, two individuals are heterozygous tall, one is homozygous dwarf. That's why the ratio here is 1:2:1. That's why the ratio here is 1:2:1. So this is about the monohybrid cross. And what Mendel concluded after this? He concluded that characters are controlled by the discrete units called. So he made certain conclusions on the basis of that, right? He made certain conclusions on the basis of that. Like if I talk about the first conclusion, and this first conclusion, okay, so as per that, factors, okay, characters are controlled by discrete units called factors, and the factors are paired. Factors are what? Factors are paired. So characters are controlled by discrete units called factors, and these factors, they are what? They are paired. Yes or no? They are paired. Yes or no? Now the second, this is the first conclusion, and on the basis of these conclusion, he gave that laws of inheritance. Now as for the second conclusion, that when, right, okay, that when two different factors, you know that now we use the term genes, right, or the alleles for the alternative forms, but at that time he used the term factors. So when two different unit factors, okay, when two different unit factors are present in an individual, only one will express itself and another will not. Yes or no? On the basis of F1 generation, he concluded it, right? On the basis of F1 generation, he concluded it that when two different factors are present in a single individual, only one, right? Only one unit factor will express itself. Only one unit factor will express itself. Yes or no? Only one unit factor will express itself, and that will be the dominant one. That will be the dominant one. I'm not writing this. So that will be the dominant one, and the one which is not, right, which is not expressed, that will be the recessive one, right? That will be the recessive one. Done, bache? Done, bache? Sure? Now the third conclusion. So these three conclusions are on the basis of monohybrid cross. So now when you talk about the third conclusion, so as per that conclusion, right, bache, that these factors, these unit factors, they do not show the blending, they do not show the mixing, right? It's not like that the tallness and the dwarfness alleles, they will get mixed, or the factors, they will get mixed. No, we can obtain them as such in the F2 generation. If you have noticed here, right, whenever we talk about the ratios, not in the case of monohybrid cross only, even in the case of dihybrid cross, when we talk about the ratios, na bache, we always check the ratio of F2 generation. Okay, we always check the ratio of what, bache? We always check the ratio of the F2 generation. Isn't it? Isn't it, Subhashini? Why? Because this law is saying that alleles do not show any blending, and both the characters are recovered as such in F2 generation. So alleles, they do not show any blending, means mixing. They do not show any blending, and both the alleles or the characters are obtained. They are obtained in F2 generation, right? Like, see, when we were talking about the first generation, we only saw the tall plants, but when we check the ratio of the F2 generation, we even saw the tall plants as well, the dwarf as well. So it is saying that allele do not show any blending. Okay, bache? Okay, bache? So this postulate, see, these two postulates, sorry, these two conclusions, the first conclusion and the second conclusion, or you can say that these two postulate, on the basis of these two postulate, the Mendel gave the law of dominance, isn't it? On the basis of these two, Mishra, Sindhura, Subhashini, everyone, please focus here. So on the basis of these two conclusions or these two postulates, Mendel gave the law of dominance. As per that law of dominance, what is there? As per that law of dominance, these unit factors, right, the characters are controlled by the unit factors. They are the discrete units. They occur in the form of a pair, and in the presence of one unit factor, the another will not express itself. The one which is expressing itself, that is the dominant one, and the one which is not expressing itself is the recessive one. Yes or no? Yes or no? So these two conclusion or postulates, they are telling us about what? They are telling us about the law of dominance, right? They are telling us about the law of dominance. Clear, bachche? Clear? Sure? Yes, are you sure? Fine. So now when it comes to the third conclusion, on the basis of that, we have the law of segregation. Which law do we have? We have the law of segregation, which is also known as the law of purity of gametes. Right, bache? It is also known as law of purity of gametes. Bache, the first law, the law of dominance is there, the law of segregation is there, and then there is the law of independent assortment. So out of these laws, this law of segregation, which is also known as the law of purity of gametes, we consider this law as a universal law. Why? Because it will always be followed. That after Mendelian inheritance, we'll be starting the post-Mendelian inheritance, and in that post-Mendelian inheritance, you will see there are deviations. There are certain, characters which do not follow these rules. Okay, so ultimately it is the law of segregation which will always be followed, but the law of independent assortment and the law of dominance will not be followed. Clear, bache? So that's why I'm saying that it is the universal law. Which law it is? It is the universal law. And as per this law, during the gamete formation, okay, during the gamete formation, see what is happening during gamete formation. Let's take the example of this capital T and small t. Of course, gamete formation will be there, because here we are discussing about the sexual reproduction. So gamete formation will be there. So during gamete formation, these unit factors, they will get separated. What is going to happen during gamete formation? During gamete formation, these factors, they will get separated. Randomly, they will get separated. Right now, they are in a pair, but at the time of gamete formation, they will get separated. They will get segregated randomly, and randomly they will move to any gamete. Okay, they will move to any gamete. And when there is the fertilization, obviously when there is the fertilization, then again that pairing will occur. But that's not mandatory that if in parent generation it is capital T, it will stay like this. That part we will discuss later. So ultimately, as per this law, that during the gamete formation, these unit factors, they segregate randomly, right? They will pass to the gametes. Fine. These unit factors, or you can say that alleles, they segregate randomly. They are passed to the different gametes, right? And each gamete will receive only one allele for a particular character. Each gamete will receive only one allele for a particular character. Let's discuss it. See, when I'm using the word unit factor, means I'm talking about the alleles. Okay, when I'm using the word unit factor, means I'm talking about the allele. Okay, so you know that Mendel used the term factor. So that's why I'm telling you like this. And these days, we call them as genes and alleles. Okay, done, Nandini? Done? So during gamete formation, the alleles or the unit factors segregate randomly and pass to the different gametes. Means when they will get separated, they will never pass to the same gamete. They will pass to the different gamete. Thus, each gamete receives only one allele for a particular character. Only one allele for a particular character, and that's why we say the gametes are pure. Like let's say we have the capital T, small t. So capital T, small t. During gamete formation, first of all, they will get separated. This capital T will pass to gamete A, small t will pass to gamete B. So there is no chance that the alleles of a same gene can pass to the same gamete. There is no chance at all. And in every condition, this law will be followed. Fine, this law will be followed in every condition. So that's why we used to say it is universal law. Okay, that's why what we used to say that this law is what? This law is universal. Is that clear? This law is what? This law is universal. Understood? Understood? So here, what did I mention? That each gamete will have only one allele for a particular gamete, right? Let's say here, I'm talking about any gamete. Let's say here you are having certain chromosomes. Fine. So when you look at the chromosome number A, capital A, small a, no chance at all that capital A and small a can pass to the same gamete. Right? There is no chance at all. There is no chance at all. So whenever you are talking about the gamete, let's say I'm giving you a sequence that these are the chromosomes present on a gamete. So tell me which sequence is possible? Which sequence is possible? Which sequence is possible? Let's say these are the sequence of the alleles in a particular gamete. So tell me which one is possible? Which one is possible? Ma'am, I have subscribed for All India Mock Test series. How to? Britika, you can join our Telegram group, right? You can join our Telegram group, and then, bache, you will find the PDF there. The syllabus is given there. Okay? Done? So, tell me which sequence is possible. Let's say these are the chromosomes. And what is this maybe? What is this maybe? Only few students are answering. What about others? Yes, so of course the first one is possible, right? A, B, C, D, F, G, H. See here, capital A and small a, both the alleles, they cannot be present on the same gamete. So this sequence is not possible. Here again, capital A, small a, right? Not possible. Here again, small a, capital B. This is also possible, right? So gametes cannot have the alleles, right? the alleles of a particular, both the alleles of a particular character. That's why bache, we say the gametes are pure for a character. Like if you are talking about this gamete, let's say A is telling us about the eye color, which is black. So obviously this gamete is having the allele for the black color only. Okay, so it is pure for that color. So right, this is the law of purity of gametes, right? This is the law of purity of gametes. Now, if you want to understand the third law of inheritance, that is law of independent assortment, but before that we need to understand the dihybrid cross. So here we will consider the inheritance of two characters at a time. Okay, what are we going to consider here? We will consider the inheritance of two characters, inheritance of two characters at a time. That's what dihybrid cross is. Clear, bache? That's what dihybrid cross is. So dihybrid. So let's say you are taking a seed which is round, shaped and yellow colored, right? Obviously it is pure parent. Then you are taking a seed which is wrinkled, shaped and green colored, wrinkled, shaped and green colored, right? That's what we have. Now when you talk about the first filial generation, right, when you are talking about what, the first filial generation, that's capital R, small r, capital Y, small y, right? Here gametes are going to be same. Now here what we need to follow, the law of, right, during gamete formation, which law do we need to follow? We need to follow always we need to follow the law of segregation or law of purity of gamete. So here, right, in the gametes, all the gametes are going to be same because it's homozygous dominant, capital R, capital Y. Here it will be small r, small y. Now look at this. Look at this. Now what we have to do, bache, let's do the selfing here. Right, let's do the selfing of the F1 individuals. Let's do what? Let's do the selfing of the F1 individuals. Capital R, small r, capital Y, small y, capital R, small r, capital Y, small y. So let's do the selfing. So before that, right, we need to focus on the gamete formation. So fork method is also there, right? Fork method is also there. That's what you can do. You can write down this capital R and small r separately. Here write capital R, small y, capital Y, sorry, capital Y, small y. So bache, these are the gametes which we are going to get. This is the fork method, right? This is the fork method. I'll give you a trick here also. So see, look at this. This parent is heterozygous dominant. This parent is also the heterozygous dominant. So when I was teaching you the law of segregation, I mentioned it very clearly that alleles segregate randomly and they will pass to the different gametes, right? Alleles, they will segregate randomly, right, bache, and they will pass to the different gamete. So each gamete will receive only one allele of a pair, right? This is what I just said. This is what I just said. So basically, capital T will pass to a different gamete, small t will pass to a different gamete, and that's not mandatory, right? That if in parent generation, like when you talk about the dihybrid cross, it's not mandatory that if in parent generation the capital R and capital Y is on the same chromosome, so the next generation will also follow the same trend. No, it can be like this, capital R and small y. This is random. This is random. Okay, so let me draw this Punnett square. Okay, let me draw this Punnett square, and then after that I will tell you about the law of independent assortment. So these are going to be the gametes here. Fine, these are going to be the gametes here. So 1, that's 2. Okay. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. And here also, right, it's same. You can see this capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Now just look at this, right, bache, you have to do the crossing. Capital R, capital Y, capital Y. See, this is the dihybrid cross. Yes or no? You please draw this cross. Everyone, please draw this cross. And you can do randomly also. Done. So now tell me, what will be the phenotype here? Quickly answer it to me. What will be the phenotype here? Yes, everyone, be quick. What will be the, Nandini, what will be the phenotype here? Tell me. Yes, obviously the genotype here is round and yellow, right? It is round and yellow. And what about this? What will be the phenotype there? What will be the phenotype there? Quick. What will be the phenotype there? Obviously round seed shape and green color. Round seed shape and green color. Yes or no? And what will be the phenotype here? Yes, please quickly revert. What will be the phenotype here? This third one. It is going to be this one, wrinkled and yellow. This one is again round and yellow. This one is again wrinkled and yellow. And here it is wrinkled and green. So basically, when you talk about the inheritance of two characters at a time in the F2 generation, right? Now you have to conclude the things. In the F2 generation, what you people are getting? Tell me. In the F2 generation, what you people are getting? You are getting non-parental combinations also, isn't it? Isn't it? What is the meaning of non-parental combinations? Tell me quickly. What is the meaning of non-parental combination? Like in the parent generation, we have round yellow, wrinkled green, and in the F2 generation, the individuals are even wrinkled yellow. The individuals are even wrinkled yellow. They are also round green. So these are non-parental combinations. These are non-parental combinations also. And when you check the phenotypic ratio here, I'm pretty sure you all are aware of this phenotypic ratio. So phenotype, when you look at the phenotype, it is going to be 9:3:3:1. 9 will be your round yellow, like the parental type. 1 will be your wrinkled green, right? 1 will be your wrinkled green, again the parental type, and 2 are non-parental type, like round green and wrinkled yellow. Done. And wrinkled yellow. So the phenotypic ratio here is 9:3:1. What is the phenotypic ratio here, bache? It is the 9:3:3:1. So what are the conclusions from this particular cross? Right? What are the conclusions from this particular cross? And bache, you can also discuss it in a different way. Right? Do you know that you can also discuss it in a different way? See, how many total number of possible phenotypes are here? 4 now? 1, 2, 3, 4. 4 different types of phenotypes are here. 4 different types of phenotypes are here. Yes or no? Yes or no? है ना? So what are the conclusions? What are the conclusions from this cross? Yes. What are the conclusions of this cross? That's very easy. Literally, that's very easy. See, as per this law, so this so as per this dihybrid cross, when inheritance of two characters, right? When inheritance of two character is considered, right? When inheritance of two characters is considered, then what is happening, bache? Tell me. When we consider the inheritance of two characters, then the segregation of one pair, is not affecting the segregation of another, right? Like when, let's say you are considering a hybrid. In that hybrid, what do you have? In simple words, let me explain this first, right? When you are considering a hybrid, what do you have? You are talking about the two characters. Let's say you are talking about the two characters. So here, right, during gamete formation, the segregation of this particular pair is not affecting the segregation of this particular pair. Means the segregation of seed shape is not affecting the segregation of the seed color. Are you getting it? The segregation of seed shape is not affecting the segregation of the seed color, right? That is the meaning of this particular cross. Okay, they are segregating independently, and in the next generation they will randomly meet. Fine. They are segregating independently, and in the next generation they will randomly meet, and this is your law of independent assortment. So as per the law of independent assortment, when you consider the inheritance of two, right, as per the law of independent assortment, when you consider the inheritance of two characters or more than two characters, right, two characters or more than two characters, bache, so at that time, see, the example is also given in NCERT. Then bache, the example is also given in NCERT. See, the same example is there. So as per this law of independent assortment, so when you consider the inheritance of two characters at a time, so the inheritance of one pair of alleles, means inheritance of that one gene is not affecting the inheritance of another. So they will randomly, right, they will independently separate from each other, segregate from each other, and randomly they will meet in next generation. And because of this law of independent assortment, bache, right, non-parental combinations are possible, right? Because of this law of independent assortment only, the non-parental combinations, they are possible. Are you getting it? Non-parental combinations, they are possible. Are you getting it? Right, bache? But this law of independent assortment, see, it is given there. This law states that when two pairs of a trait are combined in a hybrid, segregation of one pair of character is independent of the other pair of characters. Right? The segregation of one pair of character is independent of the segregation of another pair of characters. Fine? Fine? Done, bache? Sure. So this is your law of independent assortment. Okay, this is your law of independent assortment. Done, bache? So whenever there is the linkage, right, at the time of linkage, this law will not be followed. So that's why even this law is not universal. Okay, so when linkage is there, independent assortment is not there. When independent assortment is there, linkage is not there. So that's what you need to remember. Fine, that's what you need to remember. So this is all about the Mendelian inheritance. Fine, this is all about the Mendelian inheritance. So here I just told you about the inheritance of what? I told you about the inheritance of bache. Yes, the inheritance of what? The inheritance of the two genes and the one gene. So do you have any doubt here? Yes, tell me quickly. Do you have any doubt here? Quick. Sure? Okay, so I'll give you one trick here, that if you have to calculate the number of, okay, so there is a trick for you, or you can say that a kind of formula for you, that if you have to calculate that, how can you calculate the number of phenotype, number of genotype, number of phenotype, number of genotype, and number of zygotes as well, or you can say that the progeny, the offsprings. Okay, how can we do that? Do you know about it? Yes, do you know about it? Quick, that how can we check the number of phenotype, genotype, or progeny or offspring as well? Progeny or offspring as well. How can we check that? See, it's very simple. It's very simple. It's 2n, 3n, or 4n. It's 2n, 3n, and 4n. Right? No. That's not the product rule. Product rule is used to check the genotypic ratio. That's different. That's different. We will discuss the product rule here. So number of phenotype, number of genotype, and number of progeny, if you have to check, this is the formula: 2n, 3n, and 4n. Let me give you one example. Let's say if it is a monohybrid cross. Which cross it is? If it is a monohybrid cross, right? So n will be 1, 2:1. So two phenotypic ratios are there, like 3:1. That's what we have studied. 3:1. Two phenotypes are possible. Now come to the genotype. So it will be 3:1. Means 3 genotypes are possible. 1:2:1 is the ratio. 1:2:1 is the ratio. 1:2:1 is the ratio. 1 means homozygous tall. 1 means homozygous dwarf. 2 means heterozygous tall. So how many genotypes are possible? 3. N is the number of character, number of inheritance of character that we are considering. Now, number of progeny you have to calculate, it's 4n. So 4⁻1, it will be 4. It will be 4. So when you draw that Punnett square, so 4 offsprings are there. 4 offsprings are there. Let's take the example of a dihybrid cross. Let's take the example of a dihybrid cross. So when you talk about the dihybrid cross, okay, when you talk about the dihybrid cross, so 2 into 2, n is 2 here, so it will be 4. It will be 4. Phenotypic ratios are 4, means 4 possible phenotypes are there. You know that 9:3:3:1. 9:3:3:1. Clear, Sabarish? 9:3:3:1. So there are 4 phenotypes, right? Right? Round yellow, wrinkled green, round green, wrinkled yellow. So then it comes the 3:n is 2 here. 3 into 3. 3 3s are 9. I'll tell you about this also. 4 into 3. That will be 64. That is 64. Okay, sorry. My bad. Here it is 2. Yes, here it is 2. 4. N is 2 here. So 4 into 4 will be 16. So number of offsprings are 16. So look at this. 1, 2, 3, 4. So total they are 16. So that's how you can check. Okay, Priyanka, that's how you can check. And same way, in the same way you can check for the trihybrid cross. Right? In the same way you can check about the trihybrid cross. Takidi bache, we will discuss the evolution as well. Just allow me the time of a week, right? Basically from the coming week, from the Tuesday itself, we will start the classes again. Done? So now, let me tell you about the product rule. How many of you know about the product rule? How many of you know about the product rule? It's a very simple thing. See, product rule is used to check the ratio, to check the genotypic ratio. Product rule is used to check the ratio. Which ratio, Subhashini? The genotypic ratio. Let me tell you. So when you are talking about the product rule, let's say you have to check phenotypic ratio. It's very simple. Let's say you have to check phenotypic ratio. So you should know about the ratio of the monohybrid cross first. What do you need to know? You should know about the ratio of the monohybrid cross first. So when you know the ratio of the monohybrid cross, right? Mono means single, right? So see, phenotypic ratio is 3:1. Now let's say you want to check the phenotypic ratio of dihybrid cross. You can apply the product rule. How? See, 3:1, multiply it with 3:1. That's the simple mathematics. Tell me, it is the simple mathematics. So 3 into 3, 9. 3 ones are 3. Okay. 3 ones are 3 and 1. That's how you can obtain it. It's very simple. It's very simple. Okay. And purple bache, yes, it's possible to crack the NEET exam, but you have to be dedicated first. Okay. And if you are thinking to join the Avengers 3.0 batch, please do not delay that. Join it now, right? So that you can get the benefit out of it, because you will get certain leads as well, in which you can cover your backlogs also. Done? So this is about the product rule in the case of phenotypic ratio. Now let's discuss it in the case of genotypic ratio. In the case of genotypic ratio, so in the case of monohybrid cross, this is the genotypic ratio. In the case of monohybrid cross, this is the genotypic ratio. 1:2:1. So 1:2:1, multiply it with 1:2:1. Done. So 1. So that will be your genotypic ratio. So there is no need to cram. By just using this product rule, you can easily get the ratios as well. Fine. You can easily get the ratios as well. Understood? Sure? Sure? Done. So this is the product rule. So that's how you can check the ratios as well. And same you can apply for the tri-hybrid cross as well. Okay. So there is no need to cram the ratios. You can just, you can just use this product rule. Done, bache? So now let's talk about the post-Mendelian inheritance. Right, bache? Okay. Let's talk about what? Let's talk about the post-Mendelian inheritance. What are we going to discuss now? We will be discussing the post-Mendelian inheritance, that after Mendel we will see certain deviations from that basic rules. Okay. So like we'll be talking about the codominance here. First of all, we'll be talking about the incomplete dominance here. Okay, let's further divide it. So now what you have to understand, you have to now we have to learn the allelic interactions as well and the genic interactions as well. Right, now we are going to talk about what? We are going to talk about the interactions, right? Gene interactions, that's what we need to study. So that comes under this post-Mendelian inheritance, and that is what? That is the gene inheritance. Okay, bache? So that's what we need to discuss. So again. Okay, so gene, so it is a sign that I need a cup of tea, hai na? Gene interactions. So when it comes to the gene interactions, we have to discuss two things. One is your, right, bache, allelic interactions. Right, they all are the part of post-Mendelian inheritance. Here you will see what? Right, here what are you going to see? You will see, right, you will see the deviations from the Mendel's law. Right, that these inheritance, this is not going to follow. This is not going to follow what? This is not going to follow the Mendel's rule. I'll show you. One is allelic interaction and another is non-allelic interaction. One is allelic interaction and another is non-allelic interaction. Are you getting it? So when it comes to the allelic interaction, it is also known as intra, means within, intragenic interaction. It is known as what? Intragenic interaction. Now when it is non-allelic, it is known as intergenic interaction. Let me explain this part. It's very simple, students. See, allelic means you are talking about a gene. So here you will see that how two alleles are interacting with each other, right? How two alleles are interacting with each other. That's why we used to call it as intra. Intra means within. Intragenic interaction, like within a gene, how that alleles are interacting. Next is non-allelic interaction. That is your intergenic interaction, right? That how two different alleles are interacting. So when you are talking about the allelic interaction, can you tell me what are you going to study here, right? Can you tell me what are you going to study here? It's very easy. Yes, it's very easy. Tell me, it's very easy. You will be discussing about the incomplete dominance here. The codominance here. Multiple allelism. Pleiotropism. Fine, pleiotropism. And when you are talking about the non-allelic interaction, so here, bache, we are going to discuss the complementary genes, even the supplementary genes too, right? Complementary genes and epistasis. So epistasis is not given in your book, but this topic is important. So mainly we'll be talking about the epistasis here, right? What are we going to discuss mainly here? The epistasis. Okay, the epistasis. Done, bache? Done, bache? Sure? Are you sure? Yes. So mainly epistasis and the complementary genes, even the supplementary genes, additive genes, other interactions are also there. But mainly what we have to discuss this, right? Mainly what we have to discuss this. Clear, bache? So let's start with the incomplete dominance. So this incomplete dominance, this word is explaining everything. Isn't it? This word is explaining everything. So you come to this part. See, bache, as per the law of dominance, right? As per the law of dominance, what is happening? As per law of dominance, if, right, in a pair, you have two different alleles. So the allele, only one will express itself. This is the law of dominance. If in a pair of alleles, you have two different alleles, then only one is going to express itself. That's what we know, right? This is the law of dominance. But when you talk about the incomplete dominance, so that dominant allele is not expressing itself, right? What is happening here? The dominant allele, right? The dominant allele is not expressing itself completely. Let me give you one example. I'll give you, there are many examples. The example of snapdragon is there, right? Antirrhinum, also known as dog flower. Okay, the example of Mirabilis jalepa. Mirabilis jalepa, which is also known as 4 o'clock plant. These are the two famous examples. So this snapdragon is also known as dog flower. It is antirrhinum, right? And one more example is there, size of starch grain in pea plant. Okay, size of starch grain in pea plant. But a question will come from the examples, right? The question is going to come from the example. So snapdragon, as I said, it is also known as dog flower. And this anti, this is antirrhinum. So now I'll give you the example of this antirrhinum, or you can say that snapdragon flower, that actually what is happening here. See, it's very simple. Red flowers, right? You are talking about this plant now. The plant is having a red flower. When it is small r and small r, it is having white flower. So when you talk about the F1 generation, if we say that red color is dominant, right? Priya, yes, right? There is a reason that why we call it as a 4 o'clock plant, and that will be a homework for you. You are going to figure out that why do we call it as the 4 o'clock plant, right? That's going to be a homework for you. Okay, done, do it. So in the F1, right? In the F1 generation, in general, what do we discuss? In the F1 generation, in general, what do we discuss? Tell me. Done. So in the F1 generation, all the flowers should be red, but it is not like that. When it is capital R and small r, then the color is pink. So red is not completely dominating. Red is not expressing itself totally. Okay, red is not expressing itself totally, right, bache? So it is pink color. So now let's take the ratio of the F2 generation, and you know now why do we consider F2 generation? Yes, bache, why do we consider F2 generation? Because in F2 generation we can obtain another allele as well. Okay, we can obtain another allele as well. So capital R, small r, capital R, small r. So capital R, capital R, capital R, small r, capital R, small r, small r, small r. That's what we have. That's what we have. Right, bache? So capital R means red color. Capital R, small r means pink color, because it is the example of what? It is the example of incomplete dominance. And here you have the white color. Right? That's what you are going to get. Yes or no? So we have two things. Let's calculate the phenotypic ratio here, and let's calculate the genotypic ratio here. So when you look at the phenotypic ratio, see, 1 red, 2 pink, 1 white. So it is going to be 1:2, 2:1. And now at the same time, when you check the genotypic ratio, 1 is capital R, 2 are capital R, small r, and 1 is small r, small r. So even the genotypic ratio is also same. So in the case of incomplete dominance as well as in the co-dominance, the phenotypic and the genotypic ratio is same. Right? The phenotypic and the genotypic ratio is same. Okay? And even when you talk about the size of starch grain, right? When you talk about the size of starch grain, so this is the example that you need to remember. So here, the large starch grain, the small starch grain, right? And the result will be the starch grain having intermediate length. Okay, intermediate length. Done, bache? Okay, okay. Understood? Okay. Fine. So see, why is it so? Why do we see such things? Right? Why do we see such things? Why is it so that now you are getting a different trait? Right? There are many reasons for it. See, let us assume that the normal allele produces the normal enzyme that is needed for the transformation of a substrate. Right? Basically, these genes are doing what? How are they expressing themselves? By making proteins. We have already completed your chapter now. Your principles of inheritance. Yeah, we have already completed your molecular basis of inheritance, so you know about it, isn't it? How a gene is going to express itself? A gene is going to express itself by making what? By making protein. We know it very well, right? So now, let's say a gene is normal. It is making a normal enzyme which is acting on the substrate S and is transforming it, right? So theoretically, the modified allele means the mutated allele. It could be responsible for production of what? The normal or less efficient enzyme. If it is modified, it can form non-functional enzyme or no enzyme at all, right? Or no enzyme at all. So in the first case, the modified allele is equivalent to unmodified. So basically you will not see any difference then. If because of mutation, right, even still you are getting the same result. So obviously you're not going to see anything, right? The same phenotype will be there. Okay. Right. So, but if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. So, phenotype will only be dependent on the functioning of unmodified allele, right, bache? Right, which is the original one. So, basically, what are they trying to show here? They are telling us about the dominant and recessive part. So, they are just trying to explain here that one is dominant. Why? Because it is forming a kind of normal enzyme which will act on a substrate which will transform that substrate. One point. Second point here is that let's say if there is another enzyme, so if it is normal enzyme, normally produce and the normal, if a normal allele is there, will produce normal enzyme, will act on the substrate, you will get a result. If it is not there, so obviously you are not going to see the desired result. Right? On the basis of such things, on the basis of that enzymes, you are getting, you have that thing that some characters are dominant, some are not, some are recessive, right? It is basically related to the enzyme production. How is it related? It is basically related to the enzyme production. Clear, bache? Clear, bache? That's how these two things are. That's how these two things are related to each other. Clear? And now even if you are talking about the incomplete dominance, there also, right, it is related with the enzymes. It is related with that mutations, right? We are not getting that desired enzyme. Okay? Okay, so that's how you can relate these topics. Fine, bache, that's how you can relate these topics. So next topic is of co-dominance, co together, right? Here it's not like that, right? If it is a heterozygous, if we have a heterozygous, so in the heterozygous, both the alleles are going to express itself, and the best example for co-dominance is the blood group, AB blood group, the universal recipient blood group. Okay, the best example for the co-dominance is what? It is the AB blood group, bache, which is universal recipient. Clear? AB blood group, inheritance of AB blood group is the example, which is a universal recipient. And moreover, the another example here is, bache, the carrier of sickle cell anemia, right? The carrier of sickle cell anemia. This part I will explain when I will explain you the sickle cell anemia, then I will tell you. So it's HbA, normal Hb, and this one, and even the coat color in the cattle. That is also the example, right, bache? That is also the example. Understood? Understood? Like, see, look at this example, pure red, pure white, and you are getting what? You are getting the wrong color. Wrong color. So here also in co-dominance, just like the incomplete dominance, the phenotypic and the genotypic ratio is all same. The phenotypic and the genotypic ratio is all same. That is 1:2:1. Right? That is 1:2:1. Done, bache? So now I will explain you about the blood group, and then on the basis of blood group, bache, I will teach you the co-dominance as well as the next topic, that is your multiple alleles. Okay, so when you talk about the blood group, Carl Landsteiner discovered the blood group. Even in chapter body fluid and circulation also we discussed it. Nahin? We discussed it there. Carl Landsteiner explained it, right? So he gave that ABO blood grouping system. He gave that ABO blood grouping system, yes or no? Right, but he didn't mention about the, at that time he didn't mention about the AB blood group. So his student D. Castello and Sturley, right, his student D. Castello and Sturley, they explained about the AB blood group. Are you getting it? They explained about the AB blood group. Now when you talk about the blood group, obviously genes are responsible for it, isn't it? Genes are responsible for it. So I gene is going to, okay, I gene is going to control the blood group, right? I gene is responsible for that blood group. Now when you talk about this I gene is having more than one form, it is having more than two alleles, right? In general, what do we discuss? In general, Devo Bache, you will get the classes till your NEET 2024 examination. Okay, so blood group is controlled by which gene? It is controlled by I gene. Blood group is controlled by which gene? It is controlled by I gene. So this I gene is having alternative forms, and here this I gene is having more than two forms. It is having more than two forms. In general, this is what I was saying now. Yeah, you can say that more than two alleles are there. So whenever you talk about any character, which is having more than two alleles, then you will say that that character is showing multiple allelism, or that character is having multiple alleles. Are you getting it? That character is showing multiple allelism, or that character is having multiple alleles. When, right, when it is having more than two alleles. So when you talk about the blood group, these are the possibilities. So you are having three alleles for the blood group, right? So this is the multiple allelism. Now, I'll give you the example here. So now see, we have that in the pairs. Let's say when it is IA. So now let's talk about the genotype also. Right, let's talk about the genotype here also. Right, let's talk about what? Genotype. So see, this one is also dominant. This one is also dominant. But this one is recessive here. Right, this one is recessive here. What is it? This one is, yes, tell me bache, this one is recessive here. So from these blood groups, what are the possible phenotypes that we have? These 4 possible phenotypes we have. Phenotypes means, you know, like morphologically external feature. These are the 4 possible phenotypes here. Blood group A, blood group B, blood group AB, or blood group O. Yes, 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 yes. Tell me. Tell me. 4 possible, right? 4 possible phenotypes are there. But if you want to check the number of genotypes, right? You know that 4 possible phenotypes are here. That's what we know about the blood groups. But if you want to check the genotypes, so the formula here is n plus 1 over 2. What is the formula here? The formula here is n plus 1 over 2. It is n plus 1 over 2. And n is what here? N is the number of alleles here. N is the number of alleles here. Clear, bache? What is n here, Subhashini? It is the number of alleles here. So put it here in the formula. So it will be 3 plus 1 over 2. So that's going to be 6. So possible number of genotypes are 6, but possible number of phenotypes are 4 only. Possible number of genotypes are 6, but possible number of phenotypes are 4 only. Right, bache? So see, IA this. Here you can see the genotypes also. Here you can see the genotypes also. So this is the genotype, and the possible phenotype here is A, B, AB and O. Clear? A, B, AB and O. Done, bache? So here you have the 6 possible genotypes, and here you can see their phenotypes. So this is the example of what? This is the example of multiple allelism. Done, bache? This is the example of multiple allelism. Understood? Sure? Sure? Are you sure? Right? So here also you can see the same thing. Right? So this is the multiple allelism. So here you can see that AB is showing what? AB is showing the codominance. Okay? So in the blood group, you will see dominant recessive relationship, codominance also, the multiple allelism also. So you remember the basics behind it, that RBC is having the antigen, right? So that antigen will decide the blood group. Let's say if it is antigen A, the blood group will be A blood group. And in the plasma, there will be the antibodies against the B blood group. Understood? Antibodies against the B blood group. Now students, listen to me very carefully. The remaining portion, right? Remaining revision portion. Remaining revision will be done on Unacademy platform in a special class. Clear, bache? In a special class. Done? So tomorrow at 9 p.m. we will be having a special class on Unacademy platform where the remaining revision will be done. Fine. So I will try to put the link in the description box as well, but otherwise I will share it in Unacademy NEET English official Telegram group. Okay. So you have to join the link for that class, and tomorrow at 9 p.m. there will be the class where we are going to revise this principles of inheritance. I know it's Sunday and I know you people are free, so you can be the part of these classes, and that's what you have to do, bache. Okay, and if you want to join the Avengers 3.0 batch, you know what you have to do. The coupon code is Ambika10. So use that coupon code, be the part of that batch where you are going to learn from the most experienced faculties, of course, and the dedicated one and the enthusiastic one. Done? So see you all soon. So take care. Bye-bye. Thank you so much, bache. Yeah, revision class will also be there. We'll let you know about that. Right, the MCQ practice class, that where will I take? We'll update you. But as of now, I want to see you all tomorrow at 9 p.m. Right? So just subscribe to my profile. Okay, just subscribe to my profile. Okay, and you will find the classes there. Okay, you are going to find the classes there, and it is free of cost. There is nothing to pay for it. Okay, so take care. Bye bye. Thank you so much.